The discoid shape of resting platelets is maintained by a peripheral ring of bundled microtubules called the marginal band. When required to stop blood loss, activated platelets shrink their marginal band and rapidly convert to a spherical shape, a process thought to be driven by actomyosin contraction. But Karin Sadoul from the Albert Bonniot Institut in Grenoble, France, is interested in how platelet activation might be regulated by microtubule acetylation.
The marginal band is actually heavily acetylated, and we have shown previously that there's a rapid deacetylation during platelet activation. Most of the time, when microtubules are heavily acetylated, microtubule motors are important. And that's why we thought maybe microtubule motors are also important for platelet activation.
Working in the group of Sadi Koshbin, Sadoul and colleagues, led by Bubu Diagouraga, began by testing the effect of inhibiting the -n-directed motor dynein. The small molecule inhibitor, EHNA, reduced the ability of platelets to spread out on glass cover slips, indicating that platelet activation was inhibited. In contrast, inhibiting +n-directed kinesin motors had the opposite effect, inducing the formation of smaller microtubule rings even in resting platelets, and enhancing the spreading of activated platelets onto cover slips.
We tried to understand how this smaller ring structure is formed. We did a time course experiment to see how this larger marginal band shrinks to the smaller ring structure. And we were quite surprised because the marginal band didn't shrink, but actually coiled. And this induced the sphere shape of the activating platelet. So actually, kinesin inhibition resulted in two hallmarks of platelet activation: the disc to sphere shape and a smaller microtubule ring structure.
Using the state-of-the-art microscopes available at the Albert Bonniot Institute, Diagouraga et al. Investigated whether marginal band coiling also occurs during physiological platelet activation.
We tested that by looking at transiently activated platelets, which are present in freshly drawn blood. And in fact, the majority of these platelets had a coiled marginal band. So this confirmed our hypothesis that marginal band coiling could be a step during platelet activation, which is leading from the disc to the sphere shape.
Marginal band coiling could also be seen in platelets activated by various agonists, and live imaging showed that coiling occurs as the platelets convert from discs to spheres. The marginal band grows longer as it twists, and both the microtubule coiling and the platelet shape change could be reversed by treating transiently activated platelets with a dynein inhibitor.
So this suggested that dynein could slide microtubules apart and that this extends the marginal band, which will then be forced to coil due to the limited available space. And the result is the shape change of the platelets.
In resting platelets, dynein's activity would be counteracted by kinesins, maintaining the marginal band as a flat peripheral ring and keeping the platelet disc-shaped. Kinesin activity would be reduced and dynein activity increased upon transient activation, leading to marginal band coiling and a change in platelet shape. But how do activated platelets finally end up with a smaller ring-shaped marginal band?
That puzzled us a lot. We didn't see how you could come from this very coiled stage to a smaller ring structure. And actually the answer came from immunofluorescence studies where we stained long-lived acetylated microtubules and newly polymerized tyrosinated microtubules, and we observed that in resting platelets or in slightly coiled marginal bands, the two microtubule populations are perfectly co-localized. But when the curvature of the coiled marginal band became too important, the newly formed microtubules can't follow the original path anymore and will shortcut this coiled structure, and this will lead to the formation of a smaller ring structure.
Contraction of the platelet's actomyosin cortex compresses the coiled marginal band and helps newly polymerizing microtubules form a smaller ring. Inhibiting myosin with blebistatin or depolymerizing the cortex with cytochalasin D inhibited the formation of smaller rings in activated platelets. Sadoul and colleagues think that the strength of actomyosin contraction could be critical for determining whether transiently activated platelets become irreversibly activated or return to their resting state. But what do the researchers plan to investigate next?
Well, what we believe is that microtubule acetylation is important to regulate the coiling process. Microtubule motors have more affinity for acetylated microtubules and are therefore more efficient in terms of their action on these microtubules. So we think that acetylation is important to have a very quick activation of the platelets, and we have already observed that platelets from deacetylase deficient mice, which have hyperacetylated microtubules, are faster in their spreading behavior. So we would like to see now the opposite, take platelets from mice which are deficient in the acetyltransferase, which will have microtubules which are not acetylated, and see whether they will then take much longer to activate. This is one way we would like to go.
In the meantime, however, you can learn more about how motor-driven marginal band coiling is part of the platelet activation process in the paper by Diagouraga et al., published in the January 20th, 2014 issue of the Journal of Cell Biology.